Hi, my name is Pamela Coons, Associate Professor of Medicine in the Division of Oncology at Yale School of Medicine and Yale Cancer Center. I'm excited to announce ASCO's new open access journal, JCO Oncology Advances. As the inaugural editor-in-chief, I hope to support JCO Oncology Advances to become the premier platform to bridge the gap between accessible scientific research and clinical care. Stay tuned for more information, including new article types, at ascopubs.org/JCO Oncology Advances. We look forward to seeing your submissions in spring of 2024.
Hello, everyone. I'm Dave Johnson at UT Southwestern in Dallas.
And I'm Pat Loehrer from Indiana University, the Center of Global Oncology in Indianapolis.
And this is another episode of Oncology, Etc.
Yeah, before I get started, I'm going to ask you a quiz. Do you know what a myrmecologist is, Dave?
Is that somebody who studies snails and conches or no?
No, not even close.
Okay.
Do you know what a philatelist is?
I thought it was maybe a small cardiologist.
Someone who studies mermaids. No, a myrmecologist actually is a subject of this book that I just read called Scientist. It's a story about E.O. Wilson. It's a biography of him. And I didn't know that much about him.
Oh, ants.
Yes, yeah. Richard Rhodes wrote this book. It's a great read. I mean, he grew up in Alabama, Dave, so Southern Dave Johnson. At the age of 7, he was fishing. The hook got caught in his eye. He kept on fishing. He didn't seek medical attention. And later on, he ended up losing his vision in that eye. And his other eye was perfect. But he used that to focus on little things. So he loved ants and butterflies. And at the age of 18, he discovered the first colony of fire ants in the United States, eventually went to Harvard. He was instrumental in creating this encyclopedia of life, in which they're basically doing this global database for 1.9 million species. He was a bit of a controversial, but a very proactive person with conservation. He said basically that destroying the rainforest for economic gain is like burning a Renaissance painting to cook a meal. And he's talked about the Brazilian rainforest, in which less than 1% of it is now present as it was compared to 100 years ago. It's really quite amazing. His study of ants, the best quote I have from that is he said, Karl Marx was right. He said, socialism works. It's just that he picked the wrong species. But so that's my book today. But I'm going to turn it over to you, Dave, to introduce our speaker.
Well, we're very fortunate today to have David Steensma on this episode of Oncology, Etc. Dr. Steensma is a highly accomplished physician scientist and an internationally recognized expert in the diagnosis and management of myelodysplastic syndromes. He has numerous academic accomplishments ranging from characterization of the prevalence of JAK617F mutations in MDS, first description of the involvement of the IAR3 gene in human neoplasia, and more recently, the initial definition of clonal hematopoiesis of indeterminate potential, or CHIP. More about that later. Dr. Steensma is a graduate of the University of Chicago Pritzker School of Medicine. He obtained his internal medicine and his hematology-oncology fellowship training at the Mayo Clinic in Rochester. In the early 2000s, he served as a postdoctoral research fellow and visiting scholar in the molecular biology hematology laboratories of Dr. Douglas Higgs and Richard Gibson at Oxford University. He has served the medical community in many ways, including on the FDA ODAC committee, a fun activity if there ever was one, educational chair of the 2015 ASH annual meeting, and also as a member of the ABIM's Hematology Examination Committee, as well as many others. He is currently the global hematology head at Novartis Institute for Biological Research, where he oversees early phase development of malignant and non-malignant hematology drugs within Novartis. Prior to moving to industry, he served as the Edward P. Evans Chair in MDS Research at Dana-Farber Cancer Institute in Boston. He's an enthusiastic philatelist. We'll talk about that a little bit more later in the podcast. So, David, welcome to Oncology, Etc., and thank you so much for joining us.
Thank you for having me. Honored to be here.
Well, why don't we start by asking you just to tell us a little about yourself. Give us a little of your background, where you grew up, about your family, and very importantly, what and who influenced you to pursue medicine as a career.
Yeah, so my family is mostly in Michigan. My parents live there now. My sister, extended family, my wife's from there. And when the Dutch came to the U.S. during the great immigration wave around the turn of the 19th into the 20th century, a lot of them settled in Western Michigan.
Dave Johnson was there.
Watching this happen. So a lot of them settled in Western Michigan. I think it reminded them of home, flat, sandy, big body of water to the left, similar kind of climate. And that's really my origin as Dutch immigrant community on both sides. I grew up in New Jersey, though, partly because of the Vietnam War and my dad getting a low number when he was drafted and having to move there. And then they stayed there for decades. And that's where I grew up. I went to college thinking I wanted to do physics and astronomy, was fascinated by astronomy, astrophysics, thought that that was going to be my career, had a job as an undergraduate working as the observatory assistant. To this day, probably the best job I've ever had. And then I finished all the courses for my major. I took a course in cell biology just for fun. And I thought, wow, this is really interesting, and started thinking about going to medical school instead, and took organic chemistry over the summer between my junior and senior years of college. And one thing led to another. I ended up applying to medical school instead. I don't have any doctors in my family. My grandfather worked for Roche as a chemist his whole career. That's the closest we get to medicine. Previous generations. My father was a aerospace engineer. So my mom, a school teacher. You never know what pathways life is going to take you down.
Actually, I majored in mechanical engineering and I love physics, so I get it. I mean, there are answers there. Medicine's always been a little more puzzling where they create these different words that you have to memorize, but I love people who have gone through the sciences like this. But tell us a little bit, again, you've had a terrific career in academics and now you've moved to industry. Tell us a little bit about that transition. What was the attraction and what have you found to be the differences between the academics and your role now in Novartis?
I loved being an academic, and I envisioned that I would retire as such. But things changed a little bit in 2020. It was a very unsettled time. I think a lot of us thinking about what we were going to do for the next stage of our career. I was also turning 50 at the end of that year, so milestones like that tend to make us think about where are we going. And Jay Bradner, the head of Novartis's research institute, called and said, we could really use a hematologist for leading our early development. And I initially said no, I didn't think about it. Although, tremendous respect for Jay. He had been a colleague at Dana-Farber, brilliant guy. I had first met him as a medical student at University of Chicago some decades earlier. And then he called back and said, well, just come out and visit and see what we're all about. And the research institute's just based in Cambridge, Mass. So it was an easy visit. And one thing led to another. And a few months later, I was leading hematology development for Novartis. It really has been a fun transition. I think one of the big attractions for me was being able to do things at a different scale. And I love Dana-Farber, but I was really getting frustrated with all of the staffing shortages. Clinical trials were really moving slowly. And so the ability to work with a large range of centers around clinical development was very attractive. And there's a lot of great people at Novartis's research institutes, like a biotech within a larger pharmaceutical company. It's been fun for me also, not just to have that narrow focus on chip and myelodysplasia like I had before, but to be able to be involved in drug development in lymphoma and in CAR T and in sickle cell gene therapy and all kinds of other really fun programs. So one of the things I've been a little bit shocked by is actually how similar life as a early drug developer is compared to academia. I mean, the biggest difference is not seeing patients, although Novartis would have let me. Dana-Farber did not. So I've stopped seeing patients, at least for now. But there's a level of emphasis on science and on careful clinical trial development that I really appreciate. And in many ways, academia has become a little bit more corporate. Greater proportion of the funding comes from industry. There's companies that are spinning off all the time from academic institutions. I like to say my conflicts of interest declaration has gotten much easier now because there's only one company to declare compared to before it was, anyone who you'd done a trial with or an advisory board participation. So it's more similar than it is different. I think it'd be different. People have different roles within companies. And I think if I was in late stage development, really focused on what's going to be the big value return for the company, that would be a different type of job. And I'd have to think more about the financial implications and the regulatory to a greater degree than I do. I get to, work with scientists who are coming up with new molecular glue degraders and calling and saying, hey, where's the disease where this might be applicable, and talking to the chemists about engineering out different liabilities that might be an issue for our patients. And, that really is energizing.
You're one of the pioneer in CHIP. Can you talk a little bit about it and what it means to the average clinician or to the patient?
CHIP is one of the coolest discoveries in the biology of aging, I think, in the last, 20 years. And we have long had the sense that as people age, the number of hematopoietic stem cells that they have is reduced. In the late 60s, early 70s, there really seems to be a bottleneck when in childhood and young adulthood, we have thousands of different hematopoietic stem cells contributing to our blood cell production. And then there's like a colony collapse that happens. And at age 75, it's a much smaller number, 10 to 12 in most people. And some people, hematopoiesis is dominated by a single clone. But we really didn't have a understanding of the mechanism of why that happened. Until 2014, several groups, including Ben Ebert's, led by Sid Jaiswal, a young investigator who's now at Stanford, found that somatic mutations that are common in MDS and leukemia, like TET2 and DNMT3A, these are actually common in aging people with totally normal blood counts. And similar to monoclonal gammopathy of undetermined significance, there's just a portion of the older population that has these somatic mutations. And so we kind of needed a name for it. And I feel a little bit like what my mentor from Mayo, Bob Kyle, just one of my personal heroes, he had taken an observation in plasma cell disorders that Jan Waldenstrom made that was being called benign gammopathy and didn't really have a good name. And he called it MGUS, monoclonal gammopathy of undetermined significance, a name that now for more than 50 years has been part of the medical lexicon. And so we did that with CHIP. I wasn't the guy who discovered CHIP, clonal hematopoiesis. That was somebody else's discoveries, but we came up with a definition for it and a term for it, and it really took off. I mean, that paper has been cited a couple thousand times already, and it's part of the new World Health Organization classification of hematologic diseases that just debuted this summer. So that's been a fun world to be involved with, and it has clinical implications. And so with Irene Gobrial, who has a special interest in MGUS and smoldering myeloma and their progression to overt myeloma, I founded a clinic at Dana-Farber for patients with CHIP and other related precursor conditions. So the most interesting thing about CHIP, I think, is that it not only predisposes to hematologic malignancy, but because these cells, these clonal cells, circulate, they interact with the vascular endothelium in different ways. It's a risk factor for cardiovascular events. It's a risk for death from cardiac cause. It's a risk factor for worse gout, worse COPD. But it protects mechanisms that we don't fully understand yet against dementia. And we think that what's happening is these cells are getting in the brain and they are replacing some of the microglia that just undergo attrition with aging. So that's fascinating. Somatic mosaicism has been described in lots of different tissues now: the esophagus, the liver, the gut, the skin. These mutations that we normally think of as associated with malignancy can be found in many different tissues as we age. It's just part of life, it's part of the entropy of existence. But in the blood, it's special because there's no anatomical constraints on that tissue in the way that there is with a clonal proliferation in the esophagus. And so the circulation of the blood is what really makes that special.
So I'd like to go back just for a moment and talk about your corporate life, if I may. I think many, if not the majority of our listeners, come from the academic world, and so they're familiar with the day-to-day activities of an academic such as yourself, but perhaps less so with what do you do during the day as someone who is responsible for early phase drug development in industry. What does your day look like? Do you come in every morning and meet with the basic scientist, or is there a corporate meeting? How does your day-to-day activity go?
That's a great question. It's one I get a lot, actually, Dave. And I think, because all of us train in academic medical centers, it's a life that's very familiar to us. And what happens in these other arenas, government, industry, biotech, is maybe a little bit less familiar. So there's a lot of meeting that takes place, meeting with clinical trial teams, meeting with basic scientists, meeting with different operational experts and program managers. So that might involve, okay, we have a molecule that this is the safety profile of it. We have two other similar molecules that could go into the same indication. Their safety profile is a little bit different. Which one is the one that is most likely to be tolerated and beneficial for patients? It might be submitting packages for regulatory consideration by FDA and other health authorities around the world, and then responding to their feedback when they come and say, hey, we're not ready to give you this investigational new drug designation and let you start on clinical trials until you do ABC. And then we have to think about how we're going to do ABC. It can involve business development, we call it, which is, okay, this is our portfolio, but we really need an X or we really need a Y, and biotech company A has these. Are they willing to talk? And sometimes they come to us and say, hey, we've come up with this compound. We really need the power of a big company to take that forward and do a full development. Are you interested in partnering with us? I meet with my team members quite regularly in one-on-one meetings, both to talk about specific projects, but also their career development, make sure they're happy. And, just as in academia, people want to progress to different roles, that's true in the industry world too. Connecting people to others and helping them think about what's their next promotion, what's their next role going to look like. And then there's a lot of governance meetings. So these are meetings where we say, okay, this clinical trial, should we go forward with this or not? It's going to be a $100 million investment. This is the data we have to this point. Should we do this? What are the considerations? That's a big part of it. There is travel, during the COVID pandemic era, been much less so. The one thing I always heard people in industry complain about before I made this switch was the travel. There's been a lot less of that. So it's made me happy because it's been a more balanced lifestyle, that's for sure. But there is still some of that. Travel to investigative sites, travel to the big meetings like the ASCO annual meeting, EHA, the ASH annual meeting, and some smaller meetings. So it's a real mix of things.
So let me just follow up with one further question. Do you find that your reading has changed?
I think that my reading has changed a little bit, but mostly because of the breadth of programs that I am now responsible for. So in the past, I was very heavily focused, at least within medicine, on the myeloid world and trying to keep track of developments outside of that. But now I really have to pay attention to new findings in lymphoma and myeloma and non-malignant hematology to a greater extent than I was. My reading of things outside of medicine has also been a little bit broader, and there's more time for it because there's not the staying 3 hours at night and finishing your epic EMR inbox. One of the things I've really been impressed by is that in industry there's not a lot of time wasting. A lot of things that we were asked to do in academic medical centers are things that doctors really don't need to be doing. So I get to read lots of other things too.
Let me follow up on Dave's question. This is a confession on my part. There's so many things that I have done that I don't feel like I've really qualified to do. There's this imposter syndrome that we talk about. You're now the head of global hematology. Was there a sense as you're taking these steps that, wait a minute, this is really a job really too big for me and I'll try it? Or do you just have this innate confidence? How was that?
I am definitely affected by that. It was a major transition. I'd never done anything like this. And even with the assurance of knowing a number of the people at the research institute that I was going to, being confident in Jay Bradner's leadership and that of Alice Shaw, who's our translational clinical oncology leader, who's a wonderful colleague, this was still unlike anything that I'd ever done. And so, yeah, I was anxious about it. Ironically, sometimes the things that we complain about become the best preparation for what's coming in life. And about 6 years ago, Dana Farber affiliated with a community-based practice in the Brighton neighborhood in Boston called St. Elizabeth's Hospital, and provided hematology oncology services there. And they really needed somebody to go out for heme malignancies. And talk about taking me out of my comfort zone. Instead of having a very narrow clinic where I was seeing marrow failure and MDS and leukemia and MPN and CHIP patients with these conditions where I really had a comfort level, I was treating patients with HIV-associated lymphoma and myeloma and calling a lot of my colleagues back at the Farber for advice. And I grumbled about being the guy that was asked to do this. But it turned out to be terrific preparation for what I'm doing now, because my myeloma knowledge is not obsolete from fellowship 20 years ago. It's up to date. I've prescribed, a lot of the newer agents and seen what the adverse events were, and same with lymphoma, and seeing where the patient's needs were. So I'm actually really grateful for that experience now in retrospect. At the time, it felt like a nuisance. I had to leave the site and go to the other site, and I had patients in 2 hospitals, which is not easy. But I'm glad it happened.
I think that's an interesting observation. I too was at one point in my career spending time between 2 facilities with essentially a private practice that was broad and general versus the more narrow practice I had at the university. And it was challenging, but I think, like you say, it's a rewarding in many ways and helped me in my transition from being an oncologist to a chair of medicine of a major department in my later years. Well, this will wrap up part 1 of our interview with hematologist-oncologist and researcher Dr. David Steensma. In the second part of this episode, we'll ask David about an article he wrote on key opinion leaders published in the JCO a few years ago. Very provocative paper, to be sure. We'll also explore how he got into and stayed passionate about stamps, a subject about which he's written extensively. Thank you to all of our listeners for tuning in to Oncology, Etc. This is an ASCO Education podcast where we will talk about just about anything and everything. If you have an idea for a topic or a guest you'd like to hear on our show, please email us at education@asco.org.
Thank you for listening to the ASCO Education Podcast. To stay up to date with the latest episodes, please click subscribe. Let us know what you think by leaving a review. For more information, visit the Comprehensive Education Center at education.asco.org.
The purpose of this podcast is to educate and to inform. This is not a substitute for professional medical care and is not intended for use in the diagnosis or treatment of individual conditions. Guests on this podcast express their own opinions, experience, and conclusions. Guest statements on the podcast do not express the opinions of ASCO. The mention of any product, service, organization, activity, or therapy should not be construed as an ASCO endorsement.